Both standard antibiotic therapy and approved testing protocols are unreliable, and the long-term use of antibiotics have negative consequences and uncertain outcomes. Let's look at some alternative tests and therapeutic options. 28 patients with advanced Lyme disease were divided into two groups. One group was treated with TOA-free Uncaria tomentosa, cat's claw, and the control group consisted of 14 Lyme patients treated with standard antibiotic therapy. The control group experienced no significant improvements. 85% of the TOA-free treated patients no longer had B. burgdorferi positive blood tests. Dr. Luis Romero reports that 3 patients with Lyme-induced Parkinson's were 99% better after TOA-free cat's claw therapy. Larry Powers, a former Mr. America in 1962, became ill with the symptoms of Parkinson's disease in 1990. He became confined to a wheelchair and required help with eating. After learning that Lyme disease might be causing his symptoms of Parkinson's, he started taking TOA-free cat's claw. Within 3 weeks, he was out of his wheelchair and fishing for 100-pound tarpon. Scientists at the Lyme Research Group at the University of New Haven tested the in vitro effectiveness of the antibiotic doxycycline, TOA-free cat's claw, and an extract from the otaba tree called banderol against the spirochete round and biofilm forms of Borrelia burgdorferi. Our results demonstrated that both herbal agents, but not doxycycline, had very significant effects on all forms of B. burgdorferi, especially when used in combination, suggesting that herbal agents could provide an effective therapeutic approach for Lyme disease patients. In this experiment, doxycycline killed the classical spirochete form of B. burgdorferi, and the round and biofilm community's forms continued to live. Doxycycline, like minocycline, is a broad-spectrum antibiotic in the tetracycline family. Doxycycline confers neuroprotection in a 6-OHDA Parkinson model. The neuroprotective effect of doxycycline may be useful in preventing or slowing the progression of Parkinson's disease and other neurodegenerative diseases linked to glia function. In 2013, AC11, a standardized aqueous extract of Uncaria tomentosa has been shown to be neuroprotective in the roundworm model of Parkinson's disease. Patients with Lyme-induced Parkinson's have completely recovered after successful antibiotic or cat's claw supplementation, with no physical impairment or continued Parkinson's therapy. Parkinson's of unknown origin destroys the substantia nigra, and Lyme-induced Parkinson's does not, with one exception. A 63-year-old man with confirmed Lyme disease and clinical Parkinsonism died. Upon autopsy, the patient showed mild basal ganglia atrophy and substantia depigmentation. No Lewy bodies were detected. To our knowledge, this is the first report of striatonigral degeneration in a patient with B. burgdorferi infection of the central nervous system and clinical Lyme-associated Parkinsonism. Autopsy examinations of young persons dying from what appeared to be Parkinson's disease have frequently failed to confirm the basal ganglion damage that would be expected in the classic Parkinson's seen in the elderly. Parkinson's disease is a movement disorder in which the dopamine-producing brain structure, the substantia nigra, stops producing the neurotransmitter dopamine. When 50 to 80% of substantia nigra neurons die, the first symptoms of Parkinson's are noticed. If a Lyme-induced Parkinson's patient takes a DAT scan test, will their caudate and putamen look normal? If a Lyme-induced Parkinson's patient takes a colonoscopy or sigmoidoscopy, will there be evidence of Lewy bodies? 1,594 dementia patients were tested for intrathecal anti-Borrelia antibody index, a specific marker for neuroborreliosis. Of the 20, 3 had a diagnosis of Alzheimer's disease with Lewy body disease, and 1 had a diagnosis of Lewy body disease. Glutamate dysfunction has been implicated in the neurodegeneration seen in Parkinson's disease. Scientists in Taiwan tested D-cycloserine, a partial agonist at the glycine site of the NMDA receptor, in the MPTP rat model of Parkinson's disease. After MPTP injection, cycloserine improved or repaired dopaminergic degeneration, working memory, anxiety-like behavior, object recognition, and ability to navigate through a maze. These results suggest that NMDA receptors are involved in Parkinson's disease-related neuronal and behavioral dysfunctions, and that cycloserine may have clinical potential in the treatment of dementia associated with Parkinson's. D-cycloserine, brand name ceramycin, like PAS, is a second-line tuberculosis antibiotic. Ceramycin, like linezolid, is an oxazolidinone antibiotic. Ceftriaxone is a frontline antibiotic for Lyme disease and has been shown to be a possible therapeutic option for Parkinson's disease. Ceftriaxone ameliorates motor deficits and protects dopaminergic neurons in 6-OHDA lesioned rats. Effects of ceftriaxone on the behavioral and neuronal changes in MPTP-induced Parkinson's disease rat model. The beta-lactam antibiotic ceftriaxone as a treatment for the symptoms of Parkinson's disease and L-DOPA-induced dyskinesia in 6-OHDA rats. Collectively, these data indicate that ceftriaxone may be a superior treatment for Parkinson's disease than L-DOPA. The antibiotic minocycline has been shown to relieve glutamate neurotoxicity in a young woman with methamphetamine addiction and in schizophrenia patients. Adequate selenium supplementation may be an efficient strategy to prevent the detrimental glutamate toxicity, and further studies are warranted to define the therapeutic potentials of selenium in animal disease models and in humans. Anti-tumor necrosis factor treatment activates Borrelia burgdorferi spirochetes 4 weeks after ceftriaxone treatment in mice, just like anti-TNF biologics reactivated tuberculosis in patients with many different diseases. The Finnish study was remarkable in that culture and PCR were negative after ceftriaxone, but after additional treatment with anti-tumor necrosis factor alpha, viable spirochetes were recovered. The authors further concluded that negative culture and PCR cannot be relied upon as markers of treatment success. IDSA declares that there is no evidence that anyone has suffered significant chronic B. burgdorferi infection after the standard treatment. Dr. Yin Zhang believes chronic Lyme infections are caused by persister, meaning dormant non-growing L-form bacteria. The TB antibiotic pyrazinamide kills dormant M. tuberculosis, and it has been shown to have in vitro activity against B. burgdorferi. Dr. Zhang and his team are testing pyrazinamide and other compounds against L-form Lyme bacteria. B. burgdorferi can exist without iron. It lacks the genes to make iron-containing proteins and does not accumulate iron. In order to cause disease, it requires unusually high levels of manganese. Valeria Culotta, a colleague of Dr. Zhang at Johns Hopkins, is pursuing a strategy that targets B. burgdorferi manganese acquisition. The drug therapies that might emerge would target the unique metalloenzymes that bind with manganese, says Dr. Collada. Are manganese anti-knock additives to gasoline a contributing factor to the spread of Lyme disease? Allicin is the major active substance of fresh garlic and has antimicrobial activity against various bacteria, including E. coli, Salmonella, MRSA, Streptococcus, Klebsiella, Proteus, Bacillus, Clostridium, and Mycobacterium tuberculosis. Alamax Nutraceuticals has been treating Lyme patients with Alamax, a stabilized form of allicin, which is odorless. Patients receiving the combination of Alamax with a complete program of dietary and clinical controls has resulted in an 87% eradication of Lyme disease and over 90% of the co-infection. Allicin protects PC12 cells against 6-OHDA-induced oxidative stress and mitochondrial dysfunction in a Parkinson's disease model. At age 24, Ellie Lobel contracted Lyme disease, and 15 years later she moved to California for end-of-life care. Previously, she had been diagnosed with lupus, MS, chronic fatigue, and fibromyalgia. While on a short walk with her caretaker, Ms. Lobel was attacked by killer bees. Her caretaker ran for help, and Ms. Lobel could not escape being stung perhaps thousands of times. After the attack ended, she requested to be returned to her room to die. As a youth, she had an allergic reaction to a bee sting. Ms. Lobel did not die, and started to feel better and eventually made a full recovery. The toxic protein in bee venom, melittin, has been shown to completely incapacitate Borrelia burgdorferi in vitro. In addition to bee venom therapy for chronic Lyme disease, bee venom and its component, epimin, as neuroprotective agents in a Parkinson's disease mouse model. A new Lyme test has become available, the C6 Lyme peptide ELISA test. It detects 6 peptides on the surface of the spirochete, which do not evade laboratory detection like other surface antigens do. Scientists at Milford Hospital have developed a DNA Lyme test that aids clinicians in rapidly and accurately diagnosing Borrelia infections, which in turn can lead to a more timely initiation of appropriate medical treatment and improve patient outcomes. These patients represented undiagnosed cases of Lyme and related borreliosis, or persistent infection of Lyme and related borreliosis, after standard antibiotic treatments failed to eradicate the causative agents from the infected tissues. Salmonella, Helicobacter pylori, and E. coli are gram-negative bacteria, and their cell walls contain the molecule lipopolysaccharide. Clinical and pathological data suggest Parkinson's disease may result from an inflammatory process beginning in the intestinal wall that initiates alpha-synuclein aggregation, which then spreads from neuron to neuron, reaching the central nervous system. Bacteria living within the intestinal tract produce lipopolysaccharide endotoxin, a toxin known to induce Parkinsonism in animal models. By examining colon samples of Parkinson's patients obtained by sigmoidoscopy, as compared to control subjects, doctors want to describe differences in gut bacteria, assess leakiness of the gut wall, measure endotoxin and inflammation, and study characteristic Parkinson's disease pathology and evidence of inflammation. These data represent not only the first demonstration of abnormal intestinal permeability in Parkinson's disease subjects, but also the first correlation of increased intestinal permeability in Parkinson's disease with intestinal alpha-synuclein, the hallmark of Parkinson's, as well as staining for gram-negative bacteria and tissue oxidative stress. A University of Helsinki study compared the microbiota of healthy adults to that of Parkinson's patients and concluded Parkinson's patients have a different composition of intestinal bacteria. Our most important observation was patients with Parkinson's have much less bacteria from the Prevotalaceae family, unlike the control group. Another interesting discovery was that the amount of bacteria from the Enterobacteriaceae family in the intestine was connected to the degree of severity of balance and walking problems in the patients. The more Enterobacteriaceae they had, the more severe the symptoms. The Finnish study also found that in Parkinson's patients with postural instability gait difficulty, PIGD, E. coli bacteria invade the gut mucosa and are related to inflammation and oxidative stress. The patients have increased numbers of E. coli in their intestines. In another study, a PIGD motor subtype is associated with a faster rate of cognitive decline in Parkinson's disease and may be considered a risk factor for incident dementia in Parkinson's disease. In the article, Parkinson's disease-linked LRRK2 is expressed in circulating and tissue immune cells and upregulated following recognition of microbial structures. In cultured bone marrow-derived macrophages from mice, a significant upregulation of LRRK2 messenger RNA to microbial structures including lipopolysaccharide and to lentiviral particles. Lentiviral particles were derived from the Epstein Barr virus. The Epstein Barr virus, one of the most common viruses in the world, is also known as human herpes virus 4 and is the cause of mononucleosis. In the article, monoclonal antibodies against Epstein Barr virus cross-react with alpha-synuclein in human brain. The latent membrane protein 1 of Epstein Barr virus demonstrated intense immunoreactivity with Lewy bodies in Parkinson's disease, dementia with Lewy bodies, and glial cytoplasmic inclusions in multisystem atrophy. Epstein-Barr virus and Mycobacterium avium subspecies paratuberculosis peptides are cross-recognized by anti-myelin basic protein antibodies in multiple sclerosis patients. The fact that peptides from different species can be cross-recognized by antibodies targeting self-epitopes supports the hypothesis that Epstein-Barr virus and MAP might trigger autoimmunity through a common target. In the article, Viral Parkinsonism, although there is little evidence suggesting that infection by any virus directly induces Parkinson's disease, we suggest that infectious agents may be the first hit in a two-hit hypothesis, and that this insult sensitizes the brain to a later hit that might not have been pathogenic in the absence of an already primed system. When speaking of hits, the authors of Viral Parkinsonism are referring to Braak's Parkinson's disease, a dual-hit hypothesis. We propose that a neurotropic pathogen, probably viral, enters the brain via two routes, nasal and gastric, secondary to swallowing of nasal secretions in saliva. It is concluded that the most parsimonious explanation for the initial events of sporadic Parkinson's disease is pathogenic access to the brain through the stomach and nose, hence the term dual hit. In Sweden, researchers examined the relationship between sepsis and central nervous system infections and Parkinson's disease. 18,648 Parkinson's disease patients were compared with 93,240 matched controls. We found that subjects with multiple central nervous system infections, at least 5 years before the index date, had higher Parkinson's disease occurrence than those without central nervous system infections. This study provides preliminary evidence for an association between central nervous system infections, but not sepsis, and a higher future risk of Parkinson's disease. The aim of this study was to examine the association between common pathogenic infections and Parkinson's disease. 131 Parkinson's disease patients and 141 matched controls were checked for antibodies for the following pathogens: cytomegalovirus, Epstein Barr virus, herpes simplex virus, Borrelia burgdorferi, Chlamydophila pneumoniae, Helicobacter pylori. Infectious burden, bacterial burden, and viral burden were independently associated with Parkinson's disease. Serum alpha-synuclein and inflammatory cytokines, interleukin-1 beta and interleukin-6, in individuals with higher infectious burden were also significantly higher. Infectious burden of cytomegalovirus, Epstein Barr virus, herpes simplex virus, Borrelia burgdorferi, Chlamydophila pneumoniae, and Helicobacter pylori is associated with Parkinson's disease. This study supports the role of infection in the etiology of Parkinson's disease. Most bacteria have a layer of peptidoglycan in their cell wall for structural support. Peptidoglycan forms around 90% of the dry weight of gram-positive bacteria, but only 10% of gram-negative bacteria. The objective was to investigate the possible association of Parkinson's disease risk with variants in genes involved in the binding of bacterial cell wall peptidoglycan. DNA was obtained from 385 Parkinson's cases and 159 matched controls from 8 North American disorder centers. Parkinson's disease risk is associated with variants in genes that bind bacterial cell wall peptidoglycan. Common variants in pGLRP genes are associated with Parkinson's risk. Intestinal abnormalities, altered gut flora, and immune activation have all been proposed to contribute to Parkinson's pathogenesis, but information is limited. Studying peptidoglycan exposure and interaction with pGLRP variants may provide new insights into these associations. The aim of this project is to find out if LRRK2 participates in the immune response to mycobacteria. First, we will evaluate the impact of pharmacological manipulation of LRRK's activity and LRRK2 loss of function on the infective process of Mycobacterium tuberculosis in cellular models. Second, we will investigate whether the loss of LRRK2 in knockout models alters the susceptibility to infection by M. tuberculosis. At the end of the project, we will know if LRRK2 is involved in the immune response to mycobacteria. Targeting histone deacetylases, a novel approach in Parkinson's disease. In the recent past, studies with histone deacetylase inhibitors result in beneficial effects in both in vivo and in vitro models of Parkinson's disease. Various clinical trials have also been initiated to investigate the possible therapeutic potential of histone deacetylase inhibitors in patients suffering from Parkinson's disease. Butyric acid, sodium butyrate, and sodium phenylbutyrate are all histone deacetylase inhibitors. Phenylbutyrate is bacteriostatic against Mycobacterium tuberculosis and regulates the macrophage response to infection, synergistically with vitamin D3. Significant effects of oral phenylbutyrate and vitamin D3 adjunctive therapy in pulmonary tuberculosis, a randomized controlled trial. The damaging effects of short-chain fatty acids on Escherichia coli membranes. Destructive effects of butyrate on the cell envelope of Helicobacter pylori. Depletion of butyrate-producing Clostridia from the gut microbiota drives an aerobic luminal expansion of Salmonella. Reactivation of latent HIV-1 by a wide variety of butyric acid-producing bacteria. Sodium butyrate, a chemical inducer of in vivo reactivation of herpes simplex virus type 1 in the ocular mouse model. Reactivation of Epstein Barr virus lytic cycle by histone deacetylase inhibitors. Induction by sodium butyrate of cytomegalovirus replication in human endothelial cells. Reactivation of DNA viruses in association with histone deacetylase inhibitor therapy, a case series report. We report the observation of Epstein Barr virus-associated illnesses in 2 patients, and the reactivation of herpes B virus in an additional patient treated with romidepsin. These observations suggest that vigilance for DNA virus reactivation is needed to quantify risk in patients treated with histone deacetylase inhibitors.